Welcome to my half-time seminar. Biological data visualization. Biological data visualization is a branch of bioinformatics concerned with the application of computer graphics, scientific visualization, and information visualization to different areas of the life sciences. I would like to give you some background information of my PhD research. Over the past few years, we have been working with the plant gene. Plant gene, which means the Plant Genome Integrative Explorer, is a collection of interoperable web resources for searching, visualizing, and analyzing genomics and transcriptomic data for different plant species. Currently, it includes dedicated web portal for enabling in-depth exploration of poplar, Norway spruce, and Arabidopsis. In order to work with plant gene, we have to work with database queries, designing databases. We need to work with the logics, basic calculation, and algorithms. And also we have to do a lot of programming in order to develop new tools into plant gene. But today I will not going to show you anything about programming or math behind plant gene. As I mentioned earlier, we have three different websites in plant gene: Congene, PopGene, and AdGene. In each of website, we have different set of tools. For example, expression tools, genome tool, and analysis tools. Here I will show you the increasing demand of plant gene over the past 4 years. The first graph shows the PopGene usage, while the second graph shows the Congene usage. The x-axis represents the timeline, while the y-axis shows the number of page visits. The size of the bubble represents the new users coming into the plant gene. As you see here, we have continuous number of users from Sweden who are using plant gene, and also the other users from different countries are using plant gene time to time. Here I show only the top 10 users who are using plant gene. Big data or tsunami of data will require improved tools for biologists. In other words, common challenge faced by many biologists is how to benefit from data deluge without being overwhelmed by it. As a result, visualization is increasingly important in science, not because of the volume of data, but because of the complexity of the data. Biological data visualization expanded into wide area in data visualization. I selected 3 specific areas to continue my research. These are visualize genome, alignment phylogenetic, and system biology data. As a scientist, you may know how to do a research using scientific method. First, you state your questions, then do some background research, formulate hypotheses, design the experiment, then test hypotheses in order to get some results, which is quite similar to the process we have in design, which is called design process. In both processes, we don't know the end product, but we know how to get there. If we understand the both two processes, it is easy to deliver clear message to the audience. I will show you how. If we are scientists and if we create an image with lot of data, it might be confusing to the users. On the other hand, if we are a designer, if we try to make a nice figure, it could be pleasing to the audience, but we can't get the clear insight of the data using that figure. However, If we combine these two areas, we can deliver a clear message to the audience. For example, in the left side figure, we can see the clear synchrony between species. And the right side figure, we can clearly distinguish the difference between each sections. This synchrony plot I taken from the same journal, which shows the similar results. Now, I hope now you can understand the importance of data visualization in science. Here, emphasize the importance of my PhD is to help biologists to gain insight of biological data through new visualizations, tools, and methods, which are based on mainstream social science practices such as participatory design and design theory. To start with my research, I use design principle to build a tool and usability methods to evaluate the design principles. I will talk through each of these design principles and the usability methods in my coming slides. Let's look at how we use those design principles to redesigning of complex tool. Complex is a web-based tool that visualizes co-expression network of two species. This is the first version of complex tool. None of the above design principles were applied when we designed the tool. And if you try to use the tool, you may find it's difficult and complex. To start with redesigning, we started with the wireframe sketch. In design, we call it as a low fidelity prototype. Let's look at how can we apply those design principles into this wireframe sketch. Affordances, which mean the relationship between tool and the user. How the user can interact with the component. Some affordances are perceivable and some of them are not. For example, I use components similar to buttons. If you use a mouse over the component, it will be highlighted. So user can clearly understand which areas to click and which area to edit. I use same principle for other components. Signifiers. Signifiers are the signals which give clear meaning. These are signals tell you all about possible actions. For example, we give clear meaningful name to the buttons and clear error messages and validation messages. The next one is feedback. When user perform an action, he or she might need to do what's going behind the tool. So we use real-time progress bars, validation messages, and so on. Mapping. People who are working at the greenhouse know these bug herbivores are threat to their plants. So we discover this as great approach to model the real world metaphor into the plant gene. So if you find a bug in our system, you can use the same icon to contact us. So as you see, we have related bugs for each species, as well as we use poplar leaf for the popchini and spruce tree for the spruce. Then user can easily understand the principles behind the symbols. Constraints. There are several constraints that we need to consider when we develop a tool. For example, physical constraints. 8% of the men in Europe are colorblind. So we try to use colorblind safe palette when we develop our tools. Semantic constraints. We are trying to use standard representations throughout PlantGenie platform. So user can easily understand when they transition between different tools. Let's start to apply the design principles into the wireframe sketch. We did this by sitting together and apply design principle into the low fidelity prototype. As you see here, now we apply design principle. You can see the affordances, signifiers, mapping, and also the feedback. We also try to keep the consistent color, typography, and contrast throughout the website. As you see, starting from first version of complex tool to next version of complex tool, you may find second version is much pleasant and easy to use than the first version, but we need to make sure whether this is right or wrong using procedural way. So we use usability method to validate and evaluate the design principles. The first usability method is surveys. User can be anywhere in the world with completely different set of requirements. We use power of internet to enhance the conduct the usability surveys. Survey questions have been tested with selected number of users at our department to understand the relevance and complexity of the questions. Then some questions were removed or adjusted according to their suggestion. Finally, the survey was open to the entire community and results were recorded in Excel database. We created survey based on 5 usability components. Those are learnability, efficiency, memorability, error and feedback and satisfaction. All those components are based on previous literature. We started first survey from 1st of November 2015 and it went until end of December. And then we started the comparative usability survey from August 2016. It will be continuing in next few months until the end of this year. Let's look at the survey answers. One of the biggest challenges is getting people to answer to the surveys. We know we have hundreds of users are using Plangini, but 58 of them answer to the survey. This is small sample. It has lot of bias. So let's look at the survey results. As you see, most of them are plant biologists, then biologist, bioinformatician and computational biologist. By looking at the survey results based on 5 usability components, you can't get much insight from that because most of the users answered very positively. So we decided to create second survey to compare with other similar websites and identify weaknesses and improve the Plangini. As you see here, we still didn't finish the survey and we already got some positive answers by comparing with other similar sites like Python, Plaza and Thea. This is another usability method. We use website analytics to measure user behavior, usage and performances. This includes demographics, screen tracking and custom report methods. Later we utilize those analytics results to improve the Plangini usability. Here is the example result. As you see here, some parts of the site we get more user attraction while the other part of the site get less user attraction. For instance, the search input box is the starting point to the Plangini, but we still get less user attraction. While the other part like logo and projects get higher user attraction and we don't know why. We also follow the user behavioral flow throughout past years. As you see here, users started from one tool, go to another tool and go to another tool and so forth. Based on these results, we created the predefined workflows like this. User just need to start selecting workflow, then select the predefined workflow. These all workflow are based on behavioral patterns that I showed in previous slide. So user can easily test the workflow. Apart from the questionnaire, survey, and interview, we use cognitive walkthrough as a next usability methods, which means we started with the wireframe sketch, sitting together with the focus group or our group itself. We discussed the new functionality and feature that will be added to the tool. So we do this like an iterative process until we are satisfied the idea. Once we are satisfied, we directly go to the tool and implement the new function. We also use expert review as a alternative usability methods, which mean expert who are these are not biologists, but they have deep knowledge in their own field like interaction design. This is also important. If novel user comes to the site, we can understand how he can interact with the site. But the expert review, they only concern about the web design, layout, call to action, overall quality of the content, color scheme, and typography than the actual functionality. We also get feedback from our usage support. For example, here I show you quick demo. If one user need to have some question about our tool or functionality, he just need to go to our chat window and type the question. As you see here, user type, how can I download the XImage results? Then immediately we get the question into our support chat. One of our support member will assign to the conversation and he will answer to the questions, and user will immediately get the answer. In this way, we usually get the feedback from the external users, and users can also use our contact us bug reports and take a tour as alternative feedback or comments or suggestion methods. As you see here, if we do this like an iterative process, we definitely improve the plant belief usability improve as a defensive argument. But most compelling and overwhelming story is we can develop a framework that the future biologists can use using these results. So therefore, they do not need to reinvent the tool again. In Plant Genie, we have 3 different websites, PopGenie, AdGenie, and ConGenie. But there are increasing demand for external users to have their own genome browser, but we don't have time to do that. So we compiled all our Plant Genie tools into standalone software and give it to public so that they can use as their own web browser, which is called the Plant Genome Integrative Explorer Content Management System, which will be the first in-house genomic content management system to facilitate external groups in setting up their own web resources for searching, visualizing, editing, sharing, and reproducing their results. The content management system will use initial source files generated from the in-house projects. But if I say like this, you can't understand the idea of the Genie CMS. I will show you a quick case study. This is Alan. Alan has recently got his privately sequenced genome, and he would very much like to have it as a web resource similar to Plant Genie for searching, visualizing, analyzing, and editing his private genome. Unfortunately, like most of biologists, Alan is not a programmer. However, Alan's friend called Fred introduced him Genie CMS. He went to the geniecms.org website and downloaded the CMS. All he needs to do is put his raw material into the small directory in Genie CMS. Suddenly, he will get the fascinating website similar to Plant Genie for searching, visualizing, and editing his genome. Let's look at the content of Genie CMS. Genie CMS, we have Genie as a core framework, which is similar to the framework that we have in all Plangini sites. And also we have GenList, Blast and gBrowse as core plugins. Those who are using Plangini, that they know what I mean by GenList, jBrowse and Blast. And also the other tools like Ximage, Xnet and Xheatmap compiled as plugins. User can easily change the appearance of the whole web resource using customizable themes. We already installed this Genie CMS to successfully beta test users with Eucalyptus grandis genome. One is in Sweetery Technologies and other one is in South African University. If you are interested, you can download the Genie CMS in geniecms.org website and also the themes and the tools from the same website. With that, I will be working with two future projects. One is Swedish Aspen Genome Projects, which is newly resequencing data which has RNA-seq, metabolomics and phenotype data. This data will be integrated within the Plangini resource to allow their combined visualization and analysis. I also working with the Placaleden database project, which is the site was established in 1986 with the aim of assessing nutrition and water limitation in young forest in northern Sweden. With this, I thank to my two supervisors, Nathaniel Street and John Waterworth. Even though I mention I all the time, I have many people to support my work. I thank to all of them and thank you for your attention.